In this video, I'm going to talk about meiosis, which is a form of cell division that produces cells that are genetically different to the cell that it started off with half the genetic information, which is different to mitosis. I'm going to make the assumption that you've watched the mitosis video, because some of the stages within mitosis happen in here, within meiosis. And if you have watched that video, it'll make this look a little less confusing. So we have 23 pairs of chromosomes, which equals to 46 chromosomes. That's what we have, 46 chromosomes. Fruit flies, for example, have one pair, so they have 2 chromosomes. But the important thing that you need to understand is that they come in these pairs. And when you have all of these pairs, what it means is you have this, which is images of chromosomes. And chromosomes are in their pairs. So this is chromosome 1, and it's got one chromosome there and another one, chromosome 2, and so on. So you get to your 23, your 23rd pair, which is these two, the sex chromosomes. These sex chromosomes determine on what sex you are. 2 X's, so 2 of these, wouldn't mean you're a male. No, they wouldn't. They'd mean you're a female. And one of these X's and a Y, this little one here, means you're a male. So this individual who's had this done is a male. The way it works is you've got one of these chromosomes is from your mum and one from your dad. Equally, one from your mum, one from your dad, one from your mum, one from your dad. And this makes up your genetic information. So let's say eye colour is determined on chromosome 4. I don't know which chromosome it's determined on, but let's say it is chromosome 4, and if you had blue eyes, or your mum had blue eyes, or the chromosome that's been passed on from your mother has got the blue-eyed gene, and do it in a reddy brown so you can see it a bit better. Your father's chromosome was passed on, had brown eyes. This person would then have brown eyes because the brown-eyed gene speaks louder than blue-eyed gene, for all argument's sakes now. Okay. Now we have all these chromosomes, but we get each one from a sex cell. So a sex cell, like a sperm cell, still brown, would carry just dad's information. Okay, so there'd be 23 chromosomes in this, not 23 pairs. And then mum's egg, which would be much bigger than that, would also have 23 chromosomes in it, but it would have the other one, like this. And then when these two fuse together, it's going to make this 46 number. Hope that makes sense. So we have this terminology we need to think about. When you have your chromosomes in pairs, they're described as being homologous. So homologous chromosomes would be in their pair. So if we're talking about chromosome set 1, you've got one chromosome from mum, one chromosome from dad. These are in their homologous pairs. Okay. We also want to talk about how we have our sex cells. Sex cells, so the egg for the female and the sperm for the male, both have 23 when we're talking about humans, and when they come together, they make a full cell. And that full cell will have 46 chromosomes. It's described as 2n, and n meaning one set of chromosomes. So an egg has got n number of chromosomes, and a sperm cell has n number of chromosomes, and when these two come together, you get 2n, 2 sets. Something that has one set of chromosomes, that is 1n, is known as a gamete. So this is a gamete, and this is a gamete. Two gametes come together and they make a cell, a full cell with all the information. These cells that have one set of information are known as haploid. Haploid cells. So sperm is a haploid cell and an egg is a haploid cell. When two haploid cells come together, they make one like this, which is diploid. Okay? So this is just a terminology that you're going to see in meiosis. The whole point of meiosis is to start with a full cell with information from your mum. I'll draw the chromosomes as X's. Information from your mum or information from your dad, and its whole point is to go through a various number of stages that's going to make 4 daughter cells, each with half the genetic information. So there'll be 23 chromosomes in here, 23 in here, 23 in here, 23 in here. For me, I'm a male, these will become my gametes. These gametes have been created by this cell, and this cell has had its information halved. But the important thing about meiosis is that all of these cells are genetically different. And this is important because it produces variation within a population. So, if this cell broke down and everything was exactly the same in all of my sperm cells, all of my offspring are going to be the same, genetically identical. And that's not good if the human race encounters a disease. If we're all the same, we're all going to die out. But if we've all got different combinations of different genes, we are more likely to survive as a species because some will live and some won't. The ones that live may pass on the genes that it worked for it, that helped it live in that environment, and the ones that didn't won't, unfortunately. So that's why meiosis is important, because it produces this genetic diversity. It splits down one diploid cell and makes 4 genetically different haploid cells. Okay, so that's a bit of the terminology. Quite confusing when you say it like that. So let's start with meiosis and how it goes. So I'm not gonna draw 46 chromosomes. So I'll start off with my cell, my normal cell, doing its celly life. Okay, these are special cells within the gonads, and this is where they're created. Okay, so within the ovaries of female and the testicles of males. But it's got 2 sets of chromosomes, and if you've seen the video before mitosis, you'll know that chromosomes don't always look like a cross. They have just one chromatid like this. That has all the information from mum. And this one has all the information from dad. I'll just colour in a bit. So this is our cell. It has its chromosome, for argument's sake, number 1 in their homologous pairs. And this would happen for all the other 22 sets on top of this. Currently, this is 2n, because it's got all the sets from mum and all the sets from dad. The first thing that goes on in meiosis is that this cell goes through DNA replication. Goes through DNA replication, which happens in the cell cycle. Again, in my previous video, talk about that. But now the information on each chromosome doubles. So now we've got a cell that's got this information here on this chromosome is now doubled. So this is when a chromosome looks more like a cross, because it's doubled. And likewise, the male chromosome will have doubled its information. Okay? This is described as, because we had 2n before, we've still got 2n because we've got both sets from mum and dad, but now there's twice as much information, so it's 2 times 2n. This It's important to know this because sometimes you'll see it in textbooks or videos and it can get a bit confusing where these ends are coming from. What we're trying to make is one end. Okay. Next, this cell goes through its first division within meiosis. And the divisional process as it goes through is PMAT. So, prophase, metaphase, anaphase, telophase. Again, these are 4 stages that happen within mitosis. But this is known as the first division, is known as meiosis 1. So all of these are going to be called prophase 1, metaphase 1, anaphase 1, telophase 1. And the end goal of this is that you get 2 cells that are going to have these chromosomes in them, okay, but split down. So you're going to have a chromosome in 1 because it's divided, and you're going to have the other chromosome, or what's left of it, in the other. But the difference is these chromosomes through this meiosis 1 will have mixed up information. Okay, so equally, if that's got a bit of dad stuff on it, this one is going to have a bit of mum stuff on it. And that is totally random how that happens. Alright? And I'll explain this a little bit now. So, if you watch the video, this is the mitosis video, this will make sense. So, prophase 1 in cell replication is that the chromosomes condense, the centrioles go to the poles of the cell, and then the nuclear envelope breaks down. That's exactly the same as prophase 1 in meiosis 1. Okay. Okay. It's made up of these stages. But in prophase 1, an extra step takes place. And what happens is all the chromosomes line up in their homologous pairs and they come together. So this is all happening in prophase 1 whilst the nuclear envelope's breaking down and the centrioles are moving and the chromosomes are condensing. And what happens is they cross over. Okay, and this crossing over forms a structure called a chiasmata. So this is where the genetic information is exchanged. The actual name of when they're stuck together is called a bivalent. Okay, but the chiasmata is where the stuff's going on. Okay, and what happened is this information is just swapped. So some genes from this one are swapped onto that one, and some genes from this one are swapped onto that one. And this is known as independent assortment. And that is where you get this exchange of genetic material, some from mums and some from dads. And it's random, that happens in the bivalent, known as the chiasmata, and its name is independent assortment. Independent assortment. And that is where you get this exchange of information happening here, and it's called independent assortment. And it all takes place in prophase 1. Just this first bit. So that's prophase 1 here. Okay. The next difference between this PMAT and the mitosis PMAT is that in mitosis, metaphase, all the chromosomes line up. So there's 46, they all line up in the center like this. Okay? This doesn't take place in meiosis. Okay? What happens is in meiosis 1, in metaphase 1, all the chromosomes do not align in their 46 individual chromosomes, they align in their homologous pairs. So you'll actually have 23 going along here and 23 underneath. Okay, so I'll explain that again. We have this cell and they split down, okay, like it's going to happen in mitosis. And the 4 stages in mitosis are the same. They're called prophase, metaphase, anaphase, telophase. And if you haven't watched that video or you're not familiar with mitosis, please go and watch it. Prophase 1, the centrioles move to the top of the poles, the chromosomes condense, the nuclear envelope breaks down. However, these homologous pairs come together and they swap information, which means that all the chromosomes are different. Then in metaphase 1, they align up in their homologous pairs on the equator, as opposed to in mitosis, where they line up all 46. Now everything's straightforward from there. You will have your spindle fibres attached, exactly the same. And when the spindle fibres retract in anaphase 1, so when these get pulled apart, you're going to end up with, so now we're talking about anaphase 1, you're just going to end up with these different chromosomes. Now I'm drawing one of them black and one of them blue like this, but actually because of this exchange of information, they're going to look more like this. They're going to have bits of, the blue ones will have bits of black on, black ones will have bits of blue on. They're all different. So that's meiosis 1. And now we've still got 2n, because this 2 times 2n has been halved. So these cells are 2n. That's meiosis 1. Okay. We've just finished meiosis 1. Now we're going to go through another stage called meiosis 2. And in meiosis 2, you get the 4 phases again. You get prophase, metaphase, anaphase, telophase. However, this time, because geneticists are inventive, they're aptly named 2. So prophase 2, metaphase 2, anaphase 2, and telophase 2. This time, all these 4 stages take place exactly how it would in mitosis. You get, I'll draw it here quickly, you get your chromosome that's going to have a different combination of genes on it now because of what's just happened within meiosis 1. It goes through prophase 1, prophase 2, the envelope breaks down, centrioles move to the top, chromosome condenses, just like in mitosis. Then what's going to happen? So you've got your centrioles, metaphase 2, the chromosome is going to be on the middle, anaphase 2, these are going to get pulled apart, okay, and then anaphase 2, it's all going to split up into its own little cell and a membrane is going to be formed. So now what you get, you're going through all these stages, this is going to be split down into 2. This chromosome that happened before. So you're going to have 1 cell, 2 cell. But we had 2 cells, 2 products from meiosis 1. So the same happens with that. This cell goes through meiosis 2 and you end up with 4 cells each with a chromosome that's going to have a different combination of genes on it. So they're all going to have different combinations of genes because of what happened in independent assortment. These, in my instance, would now be gametes. There'll be 23 chromosomes that all look like that. And that is how gametes are created. In a female, there'd be eggs. Okay, the ovum. So let's go over that again. We'll go all the way to the beginning. We have in meiosis the production of sex cells from a normal cell that is in the gonads. Okay, so in an example with me, this happens in the testicles. You've got chromosomes, one from mum, one from dad, in all 23 pairs, and these are called homologous pairs. When you have a cell with both sets of information, one from mum and one from dad, it's known as 2N and it's known as diploid. Let's cross there for. When you have a sex cell, they have half the information from that organism, and that's known as haploid. Remember, now you've seen the process of meiosis. It's not mum's information and dad's information, it's a combination of your parents. That information's in there, okay? So, what it goes through is you have your cell that's got my chromosome from my dad, which will be a mix from my dad's mum and dad, a combination of their genes, and a chromosome from my mum, which would be a combination of her parents' genes. And depending which gene on this chromosome is dominant, is the one that I see within my phenotype, which is why you see such as blue eyes. So this one could be for blue eyes, this one could be for brown eyes. The cell goes through DNA replication, meaning there's twice as much information on all the chromosomes. Then it goes through meiosis I, which is made up of prophase 1, metaphase 1, anaphase 1, telophase 1. All the things that happen in normal mitosis, prophase, metaphase, anaphase, and the way the chromosomes move around and the spindle fibers are formed and the nuclear envelope breaks down, all the same, apart from in prophase 1, your genetic information, your homologous pairs line up, form a bivalent, and exchange genetic information through the chiasmata, depending where you're from. That makes all these different. The chromosomes have different genes on them now. They're no longer chromosome from dad and chromosome from mum. They're a combination, and it's random and it's independent assortment. It then goes through metaphase 1, where the chromosomes align along the centre, but they align along the centre in their pairs like that. Then anaphase 1 pulls them apart, telophase 1 splits a cell, and we end up with two cells with different sets of genes now. That's random. Then them two cells go through another cell division known as meiosis II, and that includes prophase 2, metaphase 2, anaphase 2, telophase 2. And all them stages are just the same as mitosis. Envelope breaks down, chromosomes align in the middle, get pulled apart, the cell membrane reforms. And then you end up with 4 genetically different cells. If this was 2N and it's been halved, this has to be N, 23 chromosomes. N, N, and N. So you've got your cell, goes through DNA replication, rep. Got one chromosome, better draw the pairs. It's now got twice as much stuff. Goes through, so that's a 2N. It's now 2 times 2N. Goes through Meiosis I, which is made up of the stages I said before. It looks like this now, but the genes are different in different orders. This each has 2n, 2n. Then it goes through Meiosis II. And we end up with 4 genetically different cells, which means that as a population, anything that sexually reproduces has different offspring and therefore better chance of survival in different environments because they've got a different combination of traits and genes that can help them survive.